In the winter of 2008, in a limestone cave tucked into the Altai Mountains of southern Siberia, a team of Russian archaeologists pulled a tiny fragment of bone out of the frozen dirt. It was a piece of a finger, no bigger than a fingernail clipping, so small and so unremarkable that at first nobody thought it would change anything. It sat in storage for 2 years before anyone ran a full genetic analysis on it. When the results came back in 2010, they didn't just surprise the scientists who ran the test. They rewrote the story of who we are. The bone did not belong to a modern human. It did not belong to a Neanderthal either, even though Neanderthal DNA had already been mapped by that point and scientists knew exactly what to look for. What they found instead was something that had never been seen before, a lineage of ancient humans that had split off from our shared family tree, hundreds of thousands of years earlier, and had never been described by a single fossil, a single tool, or a single myth. They named this new group the Denisovans, after Denisova Cave, the site where the bone was found. That discovery alone would have been enough to make headlines. But the story of Denisova Cave did not stop there. Over the following 15 years, the cave kept giving up secrets, one sediment layer at a time. Scientists eventually found not just Denisovan remains, but Neanderthal remains, modern, human remains, and even the bone of a child whose mother was a Neanderthal and whose father was a Denisovan, proof that these two separate branches of humanity had not just coexisted, but interbred. Then came an even bigger twist. 400,000 miles away, in a cave in Spain, scientists extracted DNA from bones roughly 400,000 years old, among the oldest human DNA ever recovered, and when they compared it to what they had, the genetic fingerprint pointed toward the Denisovans, not the Neanderthals everyone had assumed those bones belonged to. Suddenly, the story of ancient humans wasn't a simple two-branch tree. It was a tangled, overlapping web spanning an entire continent, and Denisova Cave sat right at the center of it. This is the story of how one tiny finger bone found in a Siberian cave forced scientists to accept the existence of a third human species, how DNA is now the primary tool for understanding our ancient relatives, and why the discoveries coming out of that single cave are still reshaping what we think we know about human origins. Let's start with the cave itself. Denisova Cave sits on the northern slopes of the Altai Mountains, near the border where Russia, Kazakhstan, China, and Mongolia nearly meet. It's not an enormous cave. It has a single large chamber with several side galleries, and it opens onto a river valley that would have offered ancient humans access to water, game animals, and shelter from the brutal Siberian winters. Locals in more recent centuries called it Ayutash, but the name that stuck came from a Russian hermit named Dionysii, who is said to have lived there in the 18th century. Archaeologists have been excavating the site since the 1970s, and for decades, most of what they found were fairly ordinary Stone Age tools and animal bones. Nobody suspected they were sitting on top of one of the most important human fossil sites on Earth. The layers of sediment inside the cave turned out to be extraordinary. Each layer represents a different span of time, some going back well over 100,000 years, and because the cave stayed cool and relatively stable, DNA inside the bones, and even in the dirt itself, was preserved far better than at most archaeological sites. This mattered enormously, because ancient DNA degrades quickly in warm, wet, or unstable environments. In a place like Denisova Cave, genetic material can survive intact for hundreds of thousands of years, locked away in bone, teeth, and even the soil where bodily fluids and tiny bone fragments broke down over millennia. When the finger bone was found in 2008, in a layer that was later dated to roughly 50,000 years old, it looked unremarkable to the naked eye. It was so small that researchers initially assumed it belonged to a young Neanderthal, since Neanderthal remains had already turned up in nearby regions of Siberia and Central Asia. The bone was sent to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, where a team led by geneticist Svante Pääbo had already built a reputation for pioneering techniques to extract and sequence ancient DNA, including the first full Neanderthal genome. Pääbo's lab ran mitochondrial DNA analysis on the finger bone, since mitochondrial DNA is passed down only from the mother and tends to survive better than nuclear DNA, making it useful for a first-pass comparison. What came back stunned the researchers. The mitochondrial DNA was different from both modern humans and Neanderthals, different enough that it suggested this individual's lineage had split off from the ancestors of Neanderthals roughly 1 million years ago. That's a huge span of evolutionary time. It meant this wasn't a slightly unusual Neanderthal or a strangely early modern human. It looked like something else entirely, a population that had been evolving on its own path for hundreds of thousands of years without anyone in the modern world knowing it existed. The team pushed further and sequenced the nuclear genome, the fuller genetic blueprint found in the cell's nucleus, which gives a much more complete picture than mitochondrial DNA alone. That analysis, published in Nature in December 2010, confirmed that the individual belonged to a population distinct from both Neanderthals and modern humans, though more closely related to Neanderthals than to us. The scientists call this new group the Denisovans, and just like that, paleoanthropology had a third named human lineage to work with, one defined not by a skull or skeleton, but almost entirely by genetic code. That's worth pausing on because it's genuinely unusual in the history of human origins research. Every other named hominin species up to that point, Homo erectus, Homo habilis, Neanderthals, had been defined by physical remains, bones, and skulls that could be measured, compared, and argued over based on shape and structure. Denisovans were different. For years after that first announcement, all scientists had to work with was a finger bone, and later a molar tooth, also found in the cave, that turned out to belong to the same population. There was no skull. There was no way to know what a Denisovan actually looked like. The species existed, as far as the world could tell, purely as a set of genetic sequences in a database. It was a human species discovered backward, defined by its genome before its face. That posed a strange challenge. How do you understand an entire vanished branch of humanity when all you have is DNA? The answer turned out to be that DNA can tell you an enormous amount, sometimes more than bones can. By comparing the Denisovan genome to genomes of modern people around the world, researchers discovered that Denisovan DNA hadn't just vanished into extinction. Traces of it were still present, quietly woven into the genomes of living populations, especially in Melanesia. People from Papua New Guinea and nearby Pacific Islands carry up to 4 to 6% Denisovan ancestry, the highest levels found anywhere in the world. That was a genuine shock. It meant Denisovans hadn't just existed in an isolated pocket of Siberia. At some point, their descendants or their genes had traveled thousands of miles, mixing with the ancestors of people who would eventually settle island Southeast Asia and Oceania. Later studies found smaller but still significant traces of Denisovan ancestry in East Asian and Native American populations, and geneticists began to piece together that there wasn't just one Denisovan population. There appeared to be at least 2, maybe 3, genetically distinct groups of Denisovans that interbred with different modern human populations at different points in time and in different parts of Asia. That single finger bone had opened a door onto something far larger and more complicated than anyone expected. An entire diaspora of a species science hadn't even known existed a few years earlier. One of the most remarkable finds to come out of Denisova Cave arrived in 2018, when researchers analyzed a bone fragment from a young girl who lived roughly 90,000 years ago. Genetic testing showed that her mother was a Neanderthal, and her father was a Denisovan. She was, in the most literal sense possible, a first-generation hybrid between 2 different human species. This wasn't a case of ancient interbreeding traced through statistical patterns across thousands of modern genomes. This was direct, physical, unmistakable proof. One individual whose 2 parents belonged to different human lineages that many scientists had assumed were geographically and reproductively separated. Researchers nicknamed her Deni. Her existence proved that Neanderthals and Denisovans weren't just distant cousins who occasionally crossed paths. In at least one documented case, they had a child together, and that cave in Siberia was the meeting point. As excavations continued, the picture got even more crowded. Researchers eventually confirmed that Denisova Cave had been home at different points across more than 250,000 years to Denisovans, Neanderthals, and modern humans. The cave's various sediment layers contained bones and stone tools attributable to all 3 groups, sometimes in layers close enough in age that scientists can't rule out the possibility that different human species lived in or near the cave around the same general period, even if not necessarily at the exact same time. A 2020 study that analyzed environmental DNA pulled directly from the cave's soil without needing an actual bone fragment found genetic traces confirming this long, overlapping occupation. Modern humans appear to have shown up in the cave's record around 45,000 years ago, meaning there's a real possibility that for some stretch of time, 3 different kinds of humans were all living in or passing through the same valley in Siberia. Then in 2021 came the discovery that pushed the Denisovan story even further back in time and forced yet another rethink. Researchers dated the oldest confirmed Denisovan fossils, a set of bone fragments recovered from deep in the cave's Stone Age layers, and found they were around 200,000 years old, making them the earliest solid evidence of Denisovans anywhere. Critically, this was also the first time Denisovan remains were found directly associated with stone tools, meaning scientists could finally start connecting Denisovan biology to Denisovan behavior and technology. Before this, it was possible to know Denisovans existed genetically without knowing much at all about how they lived, what tools they made, or how sophisticated their culture was. And that brings us to the 400,000-year-old twist, the discovery that gives this story its title. Long before anyone had heard of Denisovans, researchers had been excavating a separate cave system in northern Spain called Sima de los Huesos, the Pit of Bones. It's an extraordinary site in its own right, a nearly inaccessible underground chamber that has yielded more than 6,000 human fossil fragments, representing at least 28 individuals, all dating back roughly 400,000 to 430,000 years. Based on their skeletal structure, these individuals were originally classified as early Neanderthals, or a population closely related to the ancestors of Neanderthals, since their skulls and other bones shared many Neanderthal-like features. Then scientists managed to extract and sequence ancient DNA from one of the femurs, a technically extraordinary achievement given the age and degraded condition of the material. It became, at the time, the oldest human DNA sequence ever recovered. Researchers fully expected the genetic data to confirm what the bones already suggested, a close relationship to Neanderthals. Instead, when they compared the mitochondrial DNA to their reference samples, it lined up more closely with the Denisovan genome from Siberia than with Neanderthal DNA. This was bizarre. The Sima de los Huesos individuals looked like early Neanderthals in their bone structure, but carried a genetic signature closer to Denisovans, a population whose only confirmed fossil evidence at the time came from a cave over 4,000 miles away in Siberia. Researchers have offered a few possible explanations for this genetic mismatch, and the debate is still not fully settled. One possibility is that the Sima de los Huesos population and the ancestors of Denisovans shared a more recent common ancestor than either group shared with Neanderthals, meaning the population that eventually gave rise to Denisovans might have originally been more widespread across Eurasia before later splitting and specializing in different regions. Another possibility involves ancient interbreeding, where a wave of migration brought Denisovan-related mitochondrial DNA into a population that was otherwise developing Neanderthal-like physical traits. A third possibility is that mitochondrial DNA alone, since it only tracks the maternal line, might not tell the whole story, and that the full picture is more complicated than a single genetic marker can reveal. Whatever the precise mechanism, the discovery blew apart the simple assumption that Neanderthals stayed in the West and Denisovans stayed in the East. It suggested that during this incredibly early period of human evolution, these lineages were far more intertwined and far more geographically fluid than anyone had guessed. Put together, here's the picture that has emerged. Sometime around 500,000 years ago or more, a population of archaic humans, likely descendants of Homo heidelbergensis, or closely related group, began spreading across Europe and Asia. Over time, geographically separated populations of this group began to diverge genetically. In the West, this divergence eventually produced the Neanderthals, a population well adapted to the colder climates of Ice Age Europe. In the East, and possibly across a much wider range than currently confirmed, a related but distinct population developed into what we now call the Denisovans. These two populations were separate enough to be genetically distinguishable, yet similar enough and geographically close enough at certain points, that they interbred repeatedly, not just once, but across multiple separate encounters over tens of thousands of years. And eventually, modern humans, Homo sapiens, expanding out of Africa, encountered both of these populations, interbred with both of them too, and inherited pieces of both genomes that many people alive today still carry. That interbreeding wasn't just a genetic footnote. It had, and still has, real biological consequences. One of the most striking examples involves a gene called EPAS1, which helps regulate how the body uses oxygen at high altitude. Tibetan populations today carry a version of this gene that allows them to live and thrive at extreme elevations where oxygen levels are dramatically lower than at sea level without developing the health problems that typically come with chronic oxygen deprivation. When researchers traced the origin of this particular gene variant, they found it almost certainly came from Denisovans. In other words, an adaptation that likely evolved in an ancient human population living in the high mountains of Asia got passed down through interbreeding and is still actively helping modern Tibetan communities survive at altitudes most humans can't tolerate for long. Other studies have linked Denisovan inherited genes to variations in immune response and fat metabolism in different modern populations, suggesting that mixing with these ancient cousins gave early modern humans practical survival advantages as they spread into new and challenging environments. For a long time, one of the biggest frustrations for researchers was the total absence of a Denisovan skull. Genetics could tell you an enormous amount about ancestry, migration, and even some physical traits predicted from DNA, but it couldn't show you a face. That changed, at least partially, in 2019 when scientists re-examined a large, robust lower jawbone that had been found decades earlier on the Tibetan plateau in a cave called Baishia Karst Cave and had been sitting largely unidentified. Since the jaw itself contained no usable DNA due to its age and condition, Researchers turned to a different technique called paleoproteomics, analyzing ancient proteins preserved in the bone and tooth enamel rather than DNA. The protein analysis showed a strong match to the Denisovan lineage, making this the first confirmed Denisovan fossil found outside Denisova Cave. And, at over 160,000 years old, one of the oldest human fossils ever found on the Tibetan Plateau. It also gave researchers their first real anatomical look at what a Denisovan skeleton might have looked like, a jaw that was notably large and robust, more heavily built than a typical modern human jaw. A second Denisovan discovery, on a molar tooth found in a cave in Laos, further confirmed that these populations weren't confined to Siberia and Tibet, but had spread widely across East and Southeast Asia, adapting to environments ranging from freezing mountain plateaus to humid tropical forests. So where does all of this leave us? A single finger bone, smaller than a fingernail, found in a Siberian cave in 2008, led to the identification of an entirely new human species, defined almost entirely through genetics. That discovery then connected, through DNA comparison, to some of the oldest human remains ever found. A population in Spain that lived 400,000 years ago and had been confidently classified as early Neanderthals until their genetic code told a different story. Along the way, researchers found direct evidence of interbreeding between Denisovans and Neanderthals in the form of a hybrid child, evidence that modern humans absorbed useful survival genes from Denisovans thousands of generations ago, and evidence that Denisovans themselves weren't a small, isolated population, but a widespread and genetically diverse group stretching from Siberia to Tibet to Southeast Asia. The bigger lesson here goes beyond just Denisovans as a single case study. It's a demonstration of how completely ancient DNA analysis has transformed the study of human origins. For most of the 20th century, understanding human evolution meant digging up bones, measuring skulls, and arguing over subtle differences in shape and structure, often with very limited fossil evidence to work from. Now, scientists can extract usable genetic material from a bone fragment the size of a pebble, or even from dirt on a cave floor with no visible bones at all, and use that genetic material to identify entire populations that left behind no other physical trace. It means the fossil record, which will always be incomplete because most organisms never fossilize, and most fossils are never found, is no longer the only source of information about our past. DNA, both ancient and modern, has become a parallel archive, one that's still being read for the first time, one cave and one bone fragment at a time. It also means the story isn't over. Denisova Cave is still being excavated. New sediment layers are still being tested. Researchers are actively searching for more Denisovan fossils across Asia, hoping to find a more complete skeleton that would finally answer basic questions that are still unresolved, like exactly what Denisovans looked like, how they behaved, and how they were culturally distinct from the Neanderthals and modern humans they lived alongside and interbred with. Every population geneticist working in this field will tell you the same thing, that the human family tree isn't the simple branching diagram many of us learned in school with modern humans as a single clean line leading up from ancient ancestors. It's more like a tangled root system with different populations splitting apart, drifting into new environments, evolving distinct traits, and then coming back together again and again, exchanging genes each time they met. Beyond the bones and the DNA, Denisova Cave has also produced some of the most sophisticated artifacts ever recovered from a site this old, and they've forced researchers to rethink assumptions about who was capable of what. Among the finds pulled from the cave's Stone Age layers is a bracelet made of polished, dark green stone, chlorite, shaped with a level of precision that would normally be associated with much later, more culturally advanced periods of human history. There's also a needle carved from bird bone, delicately made and clearly designed for sewing tight, fitted clothing, the kind of garment that would have been essential for surviving Siberian winters. Beads and pendants made from animal teeth and ostrich eggshell have also turned up in layers associated with Denisovan occupation. For years, sophisticated jewelry and tailored clothing were treated as hallmarks of modern human behavior, evidence of abstract thought and cultural complexity that supposedly set Homo sapiens apart from other archaic humans. Finding these objects and layers linked to Denisovans complicates that narrative considerably. It suggests that at least one other human species was capable of similar symbolic behavior and technical skill, and that the line between us and them was far blurrier than the old story allowed. This has led some researchers to argue that Denisovans should be understood not as a primitive offshoot, but as a successful, adaptable, and in some ways remarkably capable population that thrived across an enormous and environmentally diverse range for well over 200,000 years. They survived the frozen mountains of Siberia, the thin air and extreme elevation of the Tibetan Plateau, and, based on the tooth found in Laos, the humid tropical lowlands of Southeast Asia. Few human populations, ancient or modern, have shown that kind of ecological range. It raises the very real possibility that Denisovans were, at least for a significant stretch of prehistory, one of the most widely distributed hominins on the planet, and that the reason we know so little about them isn't because they were rare or unsuccessful, but simply because so few of their remains have survived, or been found, or been recognized for what they are. That last point matters more than it might seem. Prior to the genetic testing that identified the original finger bone, there's a real chance that Denisovan remains had already been unearthed at other sites around Asia and simply misclassified, filed away in museum drawers as unidentified fragments, or lumped in with Neanderthal or early modern human material, because there was no way to tell the difference just by looking at fragmentary bone. The 2019 Tibetan jawbone is a perfect example. It had been sitting largely unstudied for decades before proteomic analysis revealed what it actually was. That means the true extent of the Denisovan world may still be sitting unrecognized in collections that have already been excavated, waiting for someone to run the right test. That 400,000-year-old link between a Spanish cave and a Siberian cave is a perfect example of just how tangled that root system really is. Two sites, thousands of miles apart, connected by DNA extracted from bones that had been sitting in the dark for hundreds of thousands of years, waiting for the technology to finally catch up and let them speak. And somewhere out there, in caves not yet excavated or fossils not yet tested, there are almost certainly more surprises waiting, more branches of the human story that haven't been told yet because the tools to read them have only existed for the last 15 years or so. Denisova Cave proved that an entire human species can be hiding in plain sight, invisible, until someone runs the right test on the right bone. And that raises an obvious question that researchers are actively chasing right now. If one finger bone rewrote the human family tree once, what else is still buried, waiting to do it again?